All right, so today we are going to be discussing protein synthesis to bring back to your recollection for what we talked about on last week with transcription and translation. Your computer should be open, but you need to turn them away from you because you won't be using them just yet. Just need them on for when we are going to be using them. You have a note sheet in front of you, which is under lesson attachments if you're watching the lesson, or if you lose your note sheet. And it obviously looks like this, and it's front and back. Your homework is to organize all of your notes. That means you need to get everything in order. Things that you're missing, you probably should start getting together because you will have a midterm when we come back from Christmas break. We have a couple of days, and then that very next week, so we have about 3 days, and then that next second week that you're back is your midterm. So we'll be starting review soon, and you will have some information that you need to review over the break as well. All right, so with that being said, we're going to go ahead and get started. All right, as we go through this, you're going to be completing your notes, and for review, actually, it's not necessarily notes, but it is review. I'm going to be expecting for you to tell me some things based on what you've learned. If you do not already have out your science notebook, I should not have to tell you to take that out. You should already have that out. So if you do not have out your science notes, you will need to take that out because I'm going to be asking you some questions. All right. So again, as we go through today's lesson, you're going to be telling me some things. You're going to be utilizing your notes and interpreting them so that you can complete this worksheet, and then you'll be doing an activity with your partner. So, what is the relationship between DNA and proteins in a cell? This question is on your sheet, but I want you to look at your notes and tell me, in your own words, what is the relationship between DNA and protein in a cell? And some of you may not even need to look in your notes. Some of you may just remember what we discussed. So how are the two related? Anybody? Tisha. Speak up, speak up.
Okay, so DNA has ribosomes, and the ribosomes are responsible for making the protein.
Okay, she says DNA has ribosomes and they're responsible for making the protein. Anyone want to add anything else to be a little bit more detailed? Okay.
The cell can synthesis is a liquid of DNA.
Okay, but what does that have to do with? You said say that one more time.
Yeah, the cell can synthesis, which is a liquid.
Okay, so but what does that have to do with protein?
It's got chromosomes inside the liquid and it's wrapped around here.
But what does that have to do with protein?
Ribosomes.
But what does that have to do with DNA? Yeah, but what does that have to do? What is the whole thing? The whole thing have to do with one another.
It's DNA decides what the protein would have.
What did we talk about as far as what DNA has to do? What have we been talking about with the cells? Do cells just stay as they are?
No.
They what? They replicate. In order for cells to replicate, what has to happen? What's the essential, the very important, necessary thing that must happen in order for that? What? What'd you say? What has to be copied? What has to be copied? The DNA has to be copied, correct? And in order for the DNA to be copied, what is the finalization of that process of DNA being copied? Protein synthesis, which is What does protein synthesis mean? Making protein. So protein synthesis is the copying of DNA. If you have that question on your paper, why is no one writing? So essentially, DNA replication is the same thing as protein synthesis. Because when DNA replicates, that creates a new cell. And creating a new cell, the major job of the cell is to make protein. These are the types of questions that's going to be asked on your midterm as well as your final exam. You should be able to develop some type of inference knowing about these two types of things. You should be able to develop a relationship. Do you need this? Can I move on? Because when you look at your notes from last week, I believe it was called DNA replication. In the whole process of DNA replication, we broke it down to build it back up, and the finalization of it was called protein synthesis. All right. The introduction for the assignment that you're going to be doing now, we have to go over some terms. The first term that you have on there is protein synthesis. What is that? The making of protein. Synthesis means to making, so there you go. Protein synthesis is the process where cells make protein. Do not write this word for word because you already wrote down your definition, the making of protein. The only thing that you should write down in parentheses is it has two phases. And then it further, what are those two phases? Transcription and translation. So phase one is transcription. So in parentheses, I will put phase one next to transcription. Translation must be phase two. I would put that. Utilizing your notes, tell me, what is transcription? What happens during transcription? DNA is copied. What else may happen, Shania? Okay, DNA is copied, but what else happens during that process? Think about my whole analogy that I gave you when going to the doctor's office.
It's not copied the exact.
Okay, it's not copied the exact same way, but what else is happening as well?
Like you said, how the doctor is pretty much taking notes and trying to.
Not the doctor, but the nurse is taking notes. And she becomes the what? She becomes the messenger. So what else happens during transcription?
DNA changes to RNA.
DNA changes to messenger RNA. So transcription is the first phase. DNA is converted to mRNA. So DNA is in the process of being copied. It's not totally copied now because that is the whole DNA replication process. That is the whole process of protein synthesis. But at this point, the DNA is converted to mRNA. This is when the zipper unzips. The DNA double helix unzips. And you'll notice that in a moment when you do your activity. And did I give you that analogy when you're zipping up your jacket and the bottom comes unzipped? Okay, so Stefan has on a perfect example. Stand for a second. His hoodie is zipped up. Now, if he misses the bottom of his hoodie or someone takes the bottom of the hoodie and unhook it, what's going to happen? It's going to, besides that. But let's say he misses the zipper. You're going to slap yourself? All right. And that unhooks. What's going to happen to that zipper? It's going to peel apart and it's going to start to separate. You all done it when you were younger and you start to have a temper tantrum and a fit because now you're stuck in your coat and you think you're going to be able to get out and your life is over. Right? You've done it. I've done it before as an adult. Like, oh man, my favorite jacket is ruined. That's how DNA happens. Thank you so much. That's how it happens. It's like a zipper, it unzips. And this is what the process of transcription is when the DNA unravels itself. And now we have to have that messenger RNA to be able to deliver the message of what's that DNA, what's that portion of nucleotides that we need to match to. Because we do understand that DNA is made up of several, what? Started with an N. Nucleotides, which consist of a sugar, phosphate, and base. And we need to know what are those other base pairs. That's what that messenger RNA's job is. Translation, this is the second phase. Now your mRNA is converted to tRNA. What does tRNA stand for? Transfer RNA. What process is this? What is happening in translation? It's interpreting. Very good. This is like going to the pharmacist and having them interpret that prescription that you cannot read. So now it's interpreting because that mRNA doesn't use ACTG. What does it use? AUCG. And DNA doesn't really recognize that. So it has to be translated to understand what that is. What does the U stand for? Uracil. And then during this process, what else happens? The final step, what else happens? Protein is made. That's what it means when it says protein molecule is a symbol. Protein is made. So now it all comes back to your memory what DNA has to do with proteins, correct? Okay. Any questions about this? I'll give you a couple more seconds to write and then I'm moving. All right. For our assignment, what you have in front of you are some cards that look like this. These are material. Your materials are transcription and translation cards. There is one set for each group of students. So therefore, go ahead and grab a set. There's 2 people in each group, with the exception of this group over here. There's 3. So grab a stack of cards. There's from A through J. You also have your protein synthesis, translation, and transcription sheet that you got from the front of the classroom. In addition to that, you have your notes. Now, what you're going to do with your partner, we're just going to look at step 1 first. With your partner, you're going to spread out the cards and in front of you on the table, and you're going to look closely to the cards. Take about 5 minutes to discuss what each card shows and how the images on the card differ. So go ahead and do step 1 and step 1 only. We'll take about 5 minutes to do that. You hear discussions. You only need 1 set for the entire group. No, you 2 are a group and you 2 are a group. Yeah. About another minute.
Oh, yeah.
I'm hearing some really good conversations. All right, now we're going to take a look at the cards and place them in the order that you think will show protein synthesis. Going from what step, what phase? Transcription through translation, which is the completion of photosynthesis, protein synthesis. All right, go ahead and take about 5 minutes to do that. All right. Not what you're supposed to see. Sorry. Come on. All right, now that you've placed them in an order that you think is protein synthesis, all this clicking of the screen to get it right. Okay. You place them in the order that you think is protein synthesis. Let's just review some things here. How does the cell make protein with the information from DNA? Take a moment, you and your partner, and describe what it is that you wrote down, or the way that you believe how cell makes protein with the information from DNA. And essentially, this is asking the same question, which was, thank you, what is the relationship between DNA and protein? Go ahead and answer this question on your paper. And if you feel like you need to make some changes to your cards, then do so then. While you're doing this. These are also the types of questions you should ask yourself when you're studying the information. About another minute. All right, group 4, what did you guys have? Stefan, get back. Hold on.
You said how the DNA, when it copies itself with another, copies itself in order to make more DNA. Protein is also DNA.
Okay. Anybody have anything else to add to that? A little bit more detail?
I say DNA replicates the copies through two phases, through transcription and translation. Completing protein synthesis is also known as making.
Okay. Anybody else have anything else? Different? Okay. So basically, when you look at this picture here, the DNA resides in the nucleus, and those are correct answers. Transcription, which you all mentioned, DNA is used as a template to make RNA. That's what transcription is, and you've already written that in your previous questions. From there, translation is the RNA is used as a template to make protein, because that translation, it has to translate what the mRNA has written down so that it can make the proper other side of the DNA. Remember, how many bases make up a what? I know that sounds crazy. 3 bases make up a amino acid. And then what? A string of protein is what? Or a protein is a what? A string of amino acids, and that's how it all works. And the protein is used by the organism. For example, enzyme could be used by the digestive system. But again, those bases code for an amino acid. That amino acid string codes for a. Those bases code for an amino acid. That amino acid string codes for a protein, which is the final objective of the system. All right. So, how does a cell make protein with the information? Basically, what we said, to make protein, a cell must convert the information contained in DNA into a messenger molecule, which is what? mRNA. Then the code messenger molecule is translated by tRNA into a string of amino acids, which make a certain protein. Make sure in your portion where you put transcription and translation, make sure the translation has something about 3 bases code for 1 amino acid. A string of amino acid codes for a protein, because that's very important. The mRNA is going to make those different bases. Those bases are going to code for an amino acid, and then that amino acid is going to code for that protein. Do not write all of this out. You need to just put something about your bases coding for amino acid and the amino acids coding for a protein. And I'm going to be moving in 1 minute so that we can move on to the next portion of the activity. All right, we good? So let's check your, I'm moving to there because you think you're writing this all down and I'm moving. Let's check your cards to see if they are correct. Actually, I'm going to freeze this for a second because I don't want to display them all at once. Naya, answer that for a second. All right, so we're going to check to see if you actually have the correct order. Give me just 1 second. Thank you. All righty. All right, for the order that you have for protein synthesis, what was your first card, letter, A through J?
D.
Say it was D or A? D is correct. What was the next? A? You put F? F is correct. Your next letters, if you don't have it correct, take a moment, put that correct, and see if you can fix it now, because some of you said A. You might need to move A somewhere else. What's next? You say A. E is next. Take a moment to kind of switch them all around. You need to fix them up. What do you think is next?
J.
J is next.
I knew that one.
Now what?
B. A.
Say B or A?
A.
A is next. This group is on the road right now. What's next?
H.
B? H? H. What's next? B. Now what?
G.
G is next.
Then C.
C. What is C doing though?
It's starting to be
Building amino acids. 3 bases make for 1 amino acid. Then you have a string of amino acids, and I coding for a protein. Very good. All right, here's your procedure. You're going to use your DNA replication notes to fill in the first row of your chart on the back. Flip it over to the back. These same instructions are on the back. The first row is called DNA replication. You're going to write down what happens. You're going to write down the subunits to make the product. I'll talk about what subunits mean in just a moment. You're going to write down where it happens. Does it happen in the cytoplasm? Does it happen in the ribosomes? Things of that nature. And then you're going to write down the end product. For the other 2 rows, transcription and translation, you are going to go to this website that is on the screen. It's also on your sheet. This is a simulation for protein synthesis. It's going to have you run through some activities for transcription so that you can fill out this information, and it's going to have you run through a short activity for translation so you could actually see it happening and write down this information. Subunits, what do I mean by that? This box is asking, is the subunit an amino acid? Or is the subunit bases? And if it's bases, which bases are there? Again, what bases have we talked about?
4.
Adenine, thymine, guanine, cytosine, and uracil.
Fantastic.
You need to make sure you write down those things. Go ahead and get started. You have about 15 minutes, and you should be able to complete it in that 15 minutes.
Yeah.